Thanks a lot, Dara, for introducing. Actually, I made the talk a bit more general, so I'll try to cover most of the topics that I did in my PhD because I just found out the talk is 45 minutes or 40 minutes, so there will be more.
Cool.
Cool. Yeah, but the general theme is about like perturbation modeling for single cells. So let me check if I can go to the next slide. Yeah. So single cell analysis recently it's been shown that it can actually be used for understanding cell fate and health and disease. And there are, let me just say I have some problem with moving between slides. Okay, so it works. Yeah, and this has been like foundation for a lot of consortia, and one of them is actually, I mean, there are like few, the Human Cell Atlas, and then a lot of data has been generated. And with single cell, we started with close to like 8 cells from 2009, and now we are at like 1 million, 1.3 by 2017, which is old now. And so by 21, I guess we have data sets with like 4.5 and up to 5 million cells. And obviously this requires large scale and robust algorithms to handle such data. And then obviously machine learning is one candidate for such analysis. And I mean, the technology evolved, and nowadays we can, I mean, it started with gene expression, just profiling transcriptomic, but nowadays we have a lot of technologies that could capture different aspects in each cell, starting from transcriptome to chromatin accessibility, to single cell DNA sequencing, TCR sequencing, so surface protein abundance capturing, so all of these things. So this allows to have like a holistic view of cellular behaviors at single cell level. And obviously, with that, there are a lot of challenges. So you have different data modalities and different data features. So obviously, the first question is how to integrate such data sets. So basically, how to integrate across different features and different data modalities. So the first computational problem which would arise from this would be multimodal data integration. So that's the first computational problem that I'll also talk about in this talk a bit. So once we have this multimodal reference atlases built, we can do like similar or like normal typical data analysis with single cell, basically annotating cells, but this time using multiple modalities. And once you have annotated the reference atlas, so you have a multiple reference atlas, you want to and obviously in healthy, you want to contrast and compare with a disease state to basically find out the differences. And so that's basically the usability of reference atlases. So once you have to integrate them and then make them usable. And finally, once you have those reference atlases, you can answer multiple questions, such as like, what is the effect of a certain drug or perturbation at single cell level? Or which gene programs have changed according to an atlas between health and disease, or which cell communication has been disrupted. So there could be like a couple of complex and like sometimes simpler questions, which could be answered by a multimodal reference atlas or generally single cell reference atlases. So I'll talk about a few work that I've done in my PhD in 3 different stages. So basically 3 different computational problems. First, integration, how to integrate multiple modalities. Second, usage. Once we have this reference atlases, how can we use them and what are the use cases actually? And so there are a few works in that direction. And finally, again, on the use cases, but this time on predicted models, once we have reference atlases plus a few perturbations, how can we leverage those perturbation atlases this time to predict the outcome of an experiment which you never saw? So I'll start with introducing the integration problem. So basically more general. As I said, this I mean, and also probably known by a lot of people in the community, this term single cell atlases is like you could basically hear it every day, but the ultimate goal is to build a tissue level atlas for each tissue, And obviously, I mean, the way that it sounds, an atlas, it should be comprehensive. So this comes with the data integration problem that you have to integrate multiple data sets from multiple sources. And this is known as, just for transcriptomic data, as data integration or batch correction problem, which is basically the, I mean, what you see here is a low dimensional representation of 4 pancreas data sets coming from 4 different labs. And I mean, if you look at the second row, the alpha cells denoted by orange color, they're different, whereas all of them are alpha cells. So obviously you would think that like they should be on the top of each other. But if you look at the first row, they are from 4 different labs. So, and the technical measurement and the technical variations basically cause them to be separate. Whereas we would like to have something like this, where all alpha cells and other cell types are on the top of each other. So if we have multiple alpha cells, they should be on the top of each other, irrespective of the source of the study. And obviously, the problem is not as simple as I said, because most of the time the biological variation is convoluted with the technical variation. But this has been a center of attention for a lot of computational method development for batch correction and data integration. And so the next slide, I'm not going to introduce another data integration method, but basically talk about once we have this atlas built and then integrated, how can we use it and make it usable, basically. So challenges in using reference atlases. First, most of these data integration algorithms are basically, they're so harsh in the sense that, and also sometimes naive in the sense that they cannot differentiate between the biological variations and technical variations. So there is always this danger that if you have a healthy and then you have a treated sample or a disease sample, and you provide them as 2 different batches for these methods, they would basically force everything to be integrated and remove the biological variation. Second, if you're working in a clinic or in a hospital or in a data generation center, and then there's like always new data sets coming, let's say from the same tissue, and then you have to integrate and rerun these algorithms every time. So this becomes challenging, and then obviously not efficient, because you always have to run these, and then this comes with like challenges of like figuring out the hyperparameters. The more data that you have, those algorithms tends to be more complicated and not easy to use. And finally, there's like the security problem and also privacy problems. If you're working in a group or like a collaboration with multiple labs, you always have to send your data somewhere to be integrated and to be sent back. So there's like also like legal and privacy challenges in a direction. So we'll try to address some of those challenges with our recent method that was recently published. And for that, we use a concept in machine learning called transfer learning. So basically, transfer learning works like you train a learning system on dataset 1, and you think that there would be some shared knowledge between dataset 1 and the dataset 2, and you would like to basically transfer this knowledge from dataset 1 and the learning system, which was trained on dataset 1 to dataset 2, in order to improve the performance on dataset 2, which is, I mean, this concept is known as transfer learning. And then we basically use this concept as transfer learning to transfer knowledge from a reference atlas to a new data set known as query. So you have a reference atlas, which you assume has been integrated by atlas builders, by Human Cell Atlas, by all these consortia. So you have like, you assume that you have such thing, which is not a strong assumption, because like these data sets are already out there. And then you have a new data set from yourself. And then you want to basically use this reference atlas to analyze your query data. So here is what we have proposed to basically make all these reference atlases usable, and using a computational framework to map a query data set on the top of reference atlases. So I'll quickly go through the sketch. So we have public reference data sets, as I said. And the first phase is basically pre-training and building these reference atlases. And this is basically model agnostic. There are a lot of deep learning models and non-deep learning models, but here we focus on a specific class of deep learning models, which showed to be, which has been shown to be basically top performer in recent data integration benchmark for single-cell genomics, single-cell transcriptomic, and generally genomics. And we use a specific class of them called conditional variational autoencoder, which they basically try to encode a gene expression profile along its label. And label here is basically the sample label or the batch label. So anything that shows us this is the study 1, study 2, or batch 1, or protocol 1, protocol 2, but not the cell type labels. And it learns a latent representation of the data, which is free of batch effect. And this is done through variable inference, which is not the focus of this talk, but we basically use the same class of models. So within scArches, which we call, which is basically short for single cell architecture surgery, we use a specific class of deep learning methods called conditional models. This could be conditional VAE or conditional GANs. So assuming that we have built a reference atlas using these methods, we can push the weights of our reference atlas into a repository or a model zoo. And then once a new data set comes, you can basically transfer the weight from the previously trained model to a new data set, which we call query, and perform transfer learning between different status like healthy and disease, or different species, or different organs, or different studies. And the way it works is we take the new data set, so basically the query data set, we add specific set of weights to the input layer of both encoder and decoder. So basically the conditional weights, which account for removing the batch effect. And we only fine tune those specific set of weights that basically leave the whole reference intact. So it doesn't change the reference representation, which makes it interpretable because you have something fixed and you want to stitch a new data set on the top. Basically, you don't want to do that. You don't want to change the reference first. Second, it's super fast because the number of variables and then like the weights that will be added here is super few. It's like just like solving a logistic regression on your laptop, and like and the scale of like basically it's not like usual deep learning parameter scale size, and which makes it fast. And yeah, so the whole thing sits in a pipeline called scArches. And I'll show you a few examples why scArches works, and then like how it works, and then why is it useful. So the first aspect is once you have the reference atlas, you want to update this reference atlas. And I'll show you with like a pancreas data sets from 5 different studies, how we build a reference atlas. So what you see is unintegrated reference atlas. So we used a conditional VAE. In this case, we showed that it works with SCVI, with TRVAE, with a normal CVAE. So all the existing architectures for single cell genomics. And then we build a reference atlas. So we remove the batch effect, we build a reference atlas, and now you assume that a new data set comes. And after running scArches, by basically adding those nodes or those weights, which we call adapters, you can integrate the new query data in the reference. And to show that if you have new cell types or new populations in the query data, and the model can basically preserve those subpopulations, we held out alpha cells, which only exists in the query and not in the reference. And then we show that basically that cell type also could be preserved, even though it hasn't been seen in the reference. And similarly, you can basically add the 5th data set and then sequentially update the reference. And yeah, so that was basically an example how it works. We showed it for more complex scenarios, such as instead of just having one tissue, you would have you can build an atlas for a whole organism, and then have like, for example, like 25 different organs or tissues here. So we built a reference mouse cell atlas called Tabula Sinis, with close to 155 different cell types and 24 different tissues. And then we projected a new data set, a query data set, which is known as Tabula Muris. And this data set contains 2 different protocols and a different time point, which were like, I guess there were like 1 year of difference between these 2 data sets, so the measurements. And then we show that you can annotate and transfer knowledge from the reference to query. So the knowledge here would be the cell type annotation, so discrete knowledge. the reference to query. And then we show that you can basically, once you have ran scArches, you can basically do this with a simple KNN algorithm, showing that the integration power is so good that you can actually use a simple classifier to transfer labels. And then we showed that like using the KNN classifier with injected with uncertainties, using simple neighborhood uncertainties, you can show that like even if a new tissue arises, you can identify the tissue and the labels that would be assigned to that tissue. Because that tissue is not merged, completely merged, basically, you can identify there are some new cell types and new tissues based on the uncertainty. As a second example, we showed that like, in addition to discrete information, you can also transfer continuous information, but this time for multimodal references. To show that, we built a multimodal reference atlas using a two side-seq experiments. So these are the experiments where we basically simultaneously measure transcriptomic plus the surface protein abundance. And so we built that reference atlas, and we projected the data set, which was only RNA-seq. Yeah. So it didn't contain any surface protein, or like basically it was not side-seq experiment, just RNA. We projected this data on the top of the reference atlas, and using the information that we had from the reference, we imputed all the missing proteins, basically the proteins that this data set didn't have, and then show that we can recapitulate and discover major cell types, such as which you can basically identify by the surface proteins, like T cells, CD8 T cells, CD4 T cells, B cells, and monocyte population. So with these two examples, I showed that like you can transfer both continuous and discrete information for single or unimodal reference atlases, and scArches is basically generalizable to both cases. But I think the most important case and the use case that we showed in this paper was that you can build a which is actually like, I guess, an ultimate goal of like organizations such as Human Cell Atlas, where you basically want to build a reference from healthy patients, and then once you have disease, and then like a disease patient, basically you want to identify what is the difference between the healthy and disease in each cell type. To basically show such application, we build a reference atlas from 3 different tissues, so from bone marrow, lung, and PBMC, and then we integrated a query data set from BAL. So this is kind of similar physiologically to, and also like cell type composition is similar to lung. And from 9 different patients. And those patients, I mean, there were like 7 patients which were diagnosed with severe and moderate COVID, and then 2 or 3 control patients. So we showed that once you have this healthy reference, you can use scArches and then project this new cohort on the top of this reference atlas. And then we went into the population that got integrated and the population that did not get integrated to basically check whether the whole thing makes sense or not. So the biggest population that we saw the biggest difference between the query and the reference were macrophages. As you can see like different clusters of severe, moderate, and controls. And then we showed that basically the model correctly identified or like integrated tissue resident alveolar macrophages. Those are the resident macrophages that exist in lung with the healthy patients. And then we could also identify 2 novel subpopulations of tissue resident alveolar macrophages. So these are basically the ones that were not in lung, and then they're basically transmitted to lung after the infection, even though we didn't have these cell types and then cell states basically in the reference. And then you can see using different markers that we have FAP-P4 positive cells, which are tissue resident cells, and then you have monocyte-derived macrophages, which is this new cell type that arise, or cell state that which basically arise in severe and moderate patients. And then we show that there's actually like a disease trajectory separating. You can see it by the expression of C1qa between this transition from TRAMs to monocyte-derived macrophages. and basically activation signals. We did it for also CD8 T cells, and then we showed that the BALF cells did not integrate. BALF and long cells, which are similar, did not integrate to PBMCs and bone marrows because they were basically different CD8 T cells. They were cytotoxic, and the naive CD8 T cells. And the reason that, and you can basically see it with CCL5, which is active and expressed in both long and BALF cells, but not in the bone marrow and PBMCs. And the reason that the patient CD8 T cells, so basically the COVID CD8 T cell, did not integrate into the reference, basically the long reference, was because they were expressing interferon signaling because they were activated. And so basically identified why the algorithm basically integrates some stuff and not integrated some stuff, and that basically makes sense. Okay, so I showed a couple of examples, but the problem with the previous model was like, I mean, it works, but you always have to do is like manual analysis and show and look at into like different genes and to see whether they actually express or not. That takes some time and then might not be always good. So we try to address that problem by incorporating by having an interpretable latent space. So instead of looking at the gene expression space, this time we actually we modified our latent space in a sense that each latent dimension encodes a specific gene module. So you can query a new data set, but you can also query a gene module and look at the representation of a reference and query in that gene module. So this is a new method. This is not still published. It will hopefully go on bioRxiv soon. We call it XPMAP. So we start with basically mining a lot of prior knowledge from databases, from articles, from experts, and converting them to readable matrices, which could be incorporated into a neural network as regularization. And you can basically repeat the same thing as you saw with scArches, but this time you could have both integration. So as you can see in panel F, you could have reference mapping with a query and reference, but you could have this reference mapping in specific gene modules. So you can look at, for example, gene module or gene programs, as we call it here. You can look at gene program 1 and see that the reference and query are integrated because they were, for example, like this gene program shows like specific cell marker or cell types that are shared between reference and query. Whereas in a second gene program, which shows, for example, like inflammation, the reference and query are not integrated. So instead of having like a global view, you have this local view that are actually interpretable because you know actually what contributes to those programs. I'll show you one example. So again, so we can build a reference atlas, this time with 4 different PBMC data sets, and we projected a data set known, I mean, you can see it with green up there. This data set contained cells from 4 patients. So 4 of them in 2 different stages, basically control, and then 4 samples with interferon beta stimulation. As you can see, the query to reference mapping works. So basically control query maps to the control reference, but you can also see this like blobs of stimulated cells from the query that did not integrate into reference. And then we basically went deep. Instead of looking at individual genes, we went into these like interpretable gene programs to see what works and what is basically the reason that these 2 integrate or not integrate. So this is because this method works it's basically built upon Bayes-Low inference. You can do differential enrichment or differential gene program analysis and find out the gene program that separate one cohort from another. And here, for example, you can see 2 gene programs that separate B cells from the rest of the cells. If you look at into those gene programs, and there are also other gene programs such as interferon signaling combined with the specific program that separate B cells that can separate B cells plus B cells in the interferon signaling. So you could see basically what separate B cells from the rest, and you could separate what and how the B cell responded to interference signaling. You can repeat this for other cell type populations. So here, another program for erythrocytes. So here you can basically see the differentiation of like progenitors, erythroid progenitors toward erythrocytes. And then you can see the trajectory in megakaryocyte development platelet gene program. And basically for other cell types such as monocytes and T cells. So instead of looking at the global view, you can look at the gene programs given by the model and analyze your data. Okay, so that was all for querying a gene program, querying a new data set, querying a gene program, but obviously that was just for like either unimodal cases or having just two modalities, but obviously this can go to arbitrary number of modalities. You still want to do reference mapping, reference building, but this time with multimodal data. So the ultimate goal is to build a reference atlas with partially overlapping data sets. So you could have like 3 modalities. So dataset 1 has 2 of them and dataset 3 has 3 of them. So basically 1 data modality is missing, but you still want to have a reference atlas. And again, you can do query to reference mapping with a data set which can contain all of the modalities in the reference or only one of them. This is basically what we desire to do. And once you have built this reference atlases, so multiple reference atlases, you can use it for data imputation and then like joint analysis of multimodal data set. So I don't go into details, but like this is the recent work that we published in an ICML workshop in CompBio, but there's going to be a journal version which comes out soon. So we formulated the problem again as variable inference, but this time we learned specific distributions for each modality, and using a specific measure, a specific way to combine this thing called product of expert, we inferred a joint distribution over different modalities. So you could have like a joint distribution over different modalities, and then we force this joint distribution for different modalities and different data set to be on the top of each other using distribution matching measures and then variational inference. So this is the general architecture. I don't go into detail, but you can have a look in the paper. I'll show you one example that like you can build a multimodal reference atlas with 2 data sets from CyTex, basically protein and transcriptomic, and then another data set which has like transcriptomic, but this time chromatin accessibility. So there are like non-overlapping modalities. You can basically build a reference atlas as before, but you can actually query new data sets from either modalities. So here we queried a paired RNA ATAC, and because the reference atlas was contained CyTex experiments, which is good to annotate small subpopulation of CD8 T cells, or basically T cells, CD8 T cells, and a subpopulation of T cells, you can transfer those granular cell type annotation from the reference to the query, which has RNA and ATAC, and those basically those modalities does not contain that much resolution to annotate T cells. So we showed that you can basically do label transfer and have more granular cell types. As a second query, just to show that this like multiple reference atlases are usable, we queried a COVID-19 data set from 198 different patients on the top of his reference. And then we show that basically it integrates, and then the reasons that, I mean, there and the parts at which it does not integrate, they're similar to the analysis they showed for the scArches. There are some reasons. Another method that we developed, but this time specifically for integrating T cell receptor sequences and transcriptomic, which was a joint work with Benjamin Schubert and led by Felix and Yang, 2 PhD students at ICB, who basically led the whole project. Again, we used multimodal deep learning and multi-view learning, but this time, since we had sequence data, we incorporated sequence-based models such as transformers, and we showed that like if you learn multimodal reference maps for TCR and single cell and transcriptomic, it can give you extra information compared to learning unimodal reference atlases learned based on TCR only or RNA only, and you can basically see it in the UMAP. So basically TCR only is you could have like this like small islands dominated by patient effects, whereas in RNA you have nice clusters. And then in the two variations that you see on the right, these are basically POE or concatenation of these models. These modalities, you could basically preserve the best of the both worlds. And then we show that on downstream tasks, such as like T-cell specificity, the joint model actually works better to predict the antibody binding and basically T-cell specificity on two different data sets compared to the unimodal models. So I'll just finish this part of my talk, and so this is going to be our hopefully cover for scArches on Nature Biotech, which basically summarized the whole first section of the part, that we want to build a reference atlas, and then we want to map a query data which can contain seen and unseen cell states, and then you basically want to analyze this reference, the query data set using the reference atlas. And the second part of the talk, I'll talk about once you have this reference atlases, and then you incorporated the disease data or like perturbed data sets into those reference atlases, you can transform them into perturbation atlases. So you could have treated and not treated cells, and basically the controlled, treated, disease. So anything that shifted the healthy state toward the perturbed states could be in this perturbation atlas. And then once you have this, and this is a sketch from our recent review on machine learning for perturbational biology, work by Yuga, a PhD in our lab, and we showed basically we reviewed like what are the tasks that you could do with such reference atlases, such perturbation atlases. We showed that like you can predict the missing measurement, basically omic measurement of a given cell state before and after a treatment or a perturbation. You could identify targets and mechanism of predictions, so basically where the drug actually hits, or perturbation interactions in a sense. In cases where you have multiple or like combinatorial perturbations, you can predict the interaction between them, or the chemical property prediction. So I'll talk about the omic measurement, and I'll show you an example from like a work which I did like 2 years ago, which we call scGen. So we got the idea from machine vision, where people showed using generative models, you can take an image of a man with basically glass, and then a man without glass. If you do this like simple arithmetic in the latent space of VAE or a GAN, you can generate an image of a woman with glass by just simply doing this simple arithmetic as you see here. So we thought that we could actually do it with cells. So once you have a, so the question is like, can we predict the perturbation effect of a cell type given observed effect in other cell types using simple arithmetics? Or do it like in cases where you have two different species. So we did scGen, and with scGen, we basically learned we wanted to predict unseen cell types after a perturbation. To do that, we encoded the gene expression using a generative model into a latent space. In that latent space, we inferred a delta vector, which basically shifts the control cells toward the perturbed states. And we added this delta vector in that low-dimensional written representation to the control cells to predict the perturbed cells. Yeah, and then we blow them back basically to the high dimensional gene expression space, where you can basically use the predicted cells. So basically it was VAE with simple latent space vector arithmetic, and the goal was to predict out of distribution or unseen cell types that we in a certain condition that you didn't see. So here's an example. We applied it on a PBMC data set before and after interferon beta stimulation. And we showed that, I mean, after a certain stimulation, you could have global responses. So basically the responses that happens everywhere, and you have cell type specific responses. And the cell type specific responses are more important. Say that you have cancer, and then like you want to identify the drugs that are actually, or you have multiple cancer and you want to find out drugs that actually work on a specific set of cancers. And so basically that drug has to have a certain specificity. So same here. So we benchmarked our algorithm by leaving out different cell types, which you can see them in the violin plot. So each time we left out, say, B cells, And then at the bottom, you see C means control, PS means predicted stimulated, and RS is real stimulated. So basically predicted and reals. And then we showed that the model captures nonlinear cell type specific effects. So, and these 10 genes that you see, they are basically ordered from top to the bottom. They are the most cell type specific responses that you see after interferon beta stimulation. So the model actually picked that up, that for some cell type, it has to upregulate CCL8 as the hardest genes, or like CXCL10, or it has not to do that. So basically finding out not responsive cells. And then we showed that you can basically do that with a linear model, but the linear model cannot capture the cell type specific effects. And then you can see this in this box plots, where we basically bin the genes according to the hardest ones, so basically the most cell type specific ones, toward the more boring responses, which are the global responses. And the biggest difference between the nonlinear models and the linear models are in the cell type specific response, which the linear models completely fail to predict. So the previous model was not end-to-end, because we had two different stages. So the training stage was different, and the inference stage was different. The latent arithmetic step was not correlated to the training. We tried to make the whole model end-to-end. So basically from input to output, everything happens in one pipeline. And then to do that, we formulated the problem of like perturbation prediction as a distribution matching problem. We basically want to shift the distribution of healthy cells toward a target distribution. And then we solved this problem by using conditional variational autoencoders, plus regularized with maximum mean discrepancy as a measure of distributional differences between two distribution, to match this distribution of like healthy and the perturbed cells. I don't go into details, but I mean, we generally show the algorithm works not just on single cells, but also on images. You can basically manipulate a specific aspect of an image, or on digits. So basically you can perform counterfactuals, like how would this image would look like after smiling, or how would that digit would look like after thinning or thickening the image. But the important part was, like, we showed that we can actually improve this end-to-end architecture, actually improves upon the non-end-to-end one compared for the cell type specific response prediction compared to STGen for the hardest genes. So on the x-axis, you see the top the beans, or like each beans contain 50 genes, and the y-axis shows the error for those genes across different cell types. And the TRVAE, the transfer VAE actually has lower error, especially in the harder genes. Okay, and then I'll talk about, I mean, the previous models were good, but like they were limited. They were just proof of concept, because the current data sets and the real challenge actually lies when you have larger screens, so a lot of data, and also in the combinatorial regimes, where basically the previous model could not model combinatorial perturbations. So we basically tried to address this. How can we improve the interpretability of the previous models? By making them interpretable, because they were basically black box. They were good for prediction, but still like not really interpretable, and also enabled him for modeling combinatorial perturbations, and also incorporating all the metadata that you could get from a typical perturbation, single cell perturbation screen, such as the patient metadata, cell type, or continuous variables such as dose and time. Yeah. So with this in mind, this is the work from my internship at Facebook AI, and a collaboration and later collaboration with them. And here's a cool movie actually made by Facebook PR team that shows what actually this model does. So let's see. So we have a cell type perturbed with two drugs. We try to learn a latent space which is free of the variation of cell types or any covariate and perturbation to get to a basal state. And then we learn to add back the perturbation effect or the covariate effects, and then basically predict the gene expression response by adding these two. Yeah, and the covariates could basically be cell types, could be patient effects, could be any metadata in discrete metadata. Yeah. And the goal is, once you have a cell type perturbed with 2 drugs or a combination of 2 drugs, and then you have cell B, which was perturbed with 3 drugs, could you basically transfer and say what would cell A would look like if it was perturbed with the 3 drugs that, or combined 3 drugs that cell B was perturbed? Yeah, to answer such counterfactual predictions. So this is the architecture. As I said, we wanted to have a interpretable model. To do that, we have to basically have linear, we have to have a learnable aspect for each metadata and each covariate. And in the architecture, you can see that we encode the gene expression, we get to this basal state, which is we remove the variation that comes from perturbation or covariates using adversarial training. So basically this goes into 2 discriminators. And we learn a compositional generative process in the latent space. So we basically try to add back the effect of covariates and latent spaces by learning embeddings for each, and scaling the perturbations and the covariates with continuous encoder, which basically encodes the time or dosage of a certain drug. So we learn and add all of them back, and then we predict the gene expression. So this model can be used to predict novel conditions, so novel combinations that we're not measuring in the training. Since we also explicitly model dose response, we can have dose response analysis. And since we actually learn the covariate and the perturbations, we can have latent perturbation effect and latent covariate effects, which basically can compare the similarity of 2 different perturbations. I think I have like 5 minutes. Is it correct, Laura? Or
yeah.
Okay, so I'll try to basically be faster a bit. So we applied the model on a, I think this is the largest single cell perturbation screen from cold trap in the lab with 3 different cell lines and then 188 drugs at 4 different dosages. So overall, we had like 500,000 cells and then 4,000 unique conditions. So this is basically a future single cell high throughput screen now. I mean, they wanted to basically show off their multiplexing technology, but we actually applied the model on this data set. And then we showed that if you leave out a drug on a specific cell line, you can predict that, but I'll try to be faster. But most important part, since we had, we basically explicitly modeled the dose responses, the model can actually pick up whether a certain cell line is responsive at different dosages to a drug or not. So it basically picked out this afloxacin drug is, so that this cell line is not responsive to this at different dosages of this drug, but is responsive to the other 2 drugs. And more importantly, since these different cell lines were super different, the model actually picked up latent space, which shows that there is no similarity between them because they were from different cancer cell line, but it picked up the similarity of the perturbations or drugs inducing similar gene effects. So you could see that this is basically the drug latent space inferred by the model, and it showed that basically there are, like, you can see 3 different clusters, and the rest of the drugs were not really responsive. But these 3 clusters showed similar drug with similar mode of action, which basically says that these drugs had similar gene expression effect. And such maps could be actually used for repurposing efforts to use 2 different drugs that actually induce the same gene expression effect. And then we basically validate this by applying the model on L1000 datasets or links, and then show that similar drugs actually clustered together in bulk data too. And finally, we applied the model on a combinatorial CRISPR screen with 287 single and double knockouts, and then we showed that the model can capture the genetic manifold of this CRISPR knockouts. And you can basically find out different gene knockouts or combination of knockouts that induce similar gene programs. And then we identify those gene programs. But more importantly, we show that if the model hasn't seen a certain gene combination or double knockout, for example, the synergistic interaction, it can actually predict those synergistic interactions. Similarly, for epistatic interactions, if you have one knockout basically dominates the other effects, so masking effects, the model can actually pick that up too. But more importantly, we showed that because these CRISPR screens are expensive, so you cannot do it for all the knockouts that you have in mind. So also for this experiment, like out of like 6,000 possible combinations, they only measured like 100 of them, and then 5,000, like 5,000 something of them, like were not measured. Using this model, we predicted all missing combinations, all 5,600 different combinations, and then we showed that using uncertainty that comes from the model, you can identify what are the main driver for each missing combination. So if you have combination A with all other combinations, we basically showed that like most of the cluster that you can recover from this perturbation latent space, they are dominated by one, but they're all also like different effects. So here is like the gene expression of the predicted and some reals for KL of 1. So this is like an overexpression experiment, and for other genes. And then for like all the predicted values, we showed that like there could be different behaviors such as epistasis, dominant, synergy, although the model never saw those effects. And obviously the model has some failures. So if there are like new morphic or like completely new response arises and what there was not in the data set, it cannot predict that. And I'll skip this one but generally we showed that it also works across to basically translate the effect across species. And but I'll refer you to the paper. And with this, I'll just have a short outlook. This perturbation effect modeling is just super interesting direction. And then you can basically also expand this toward multimodal. So you have interventions, could be drugs, could be genetics, but you can also predict the effect of such perturbations on multiple modalities, not just gene expression, but for example, for images or for like spatial dependencies that would change after a certain, and there are a lot of machine learning problems that arise from multitasking, also experimental design and active learning, and it's also closely related to domain generalization and uncertainty estimation. So a lot of computational and machine learning problems actually arise for this. And also causal modeling, because all of these counterfactuals could be like causal interventions, obviously. Another approach would be knowledge-based approaches, not just using gene expression, but also similar to the approaches that I showed, that you can actually incorporate knowledges. Here you can also incorporate pathways or like different knowledges from papers or databases for perturbation modeling. So with this, I'd like to thank all the collaborators and people who help or led the studies that I showed from Ty's lab and also Facebook AI and other labs. Yeah, I'll be happy to take questions. Thanks a lot for listening.